so much to the organizers for inviting me to this wonderful workshop. I learned a lot, and I don't need really to sort of provide a big introduction for the topic of my talk, as I usually do when talking to fellow neuroscientists. I have to remind them that we live in a 3D world, and sort of with, perception, action, memory is affected by the fact that we live in a three-dimensional world. I definitely don't need to convince people in this room about this. And I'm going to focus on one specific aspect of coding in neural networks of three-dimensional variables, like the heading of self in 3D. Towards the end of the talk, I will also sort of touch upon issues related to how we encode the orientation of objects in 3D with respect to ourselves. This is work that has been done by Hervé in my lab now. He has his own lab in the Center for Theoretical Physics in Marseille, and Alon Rubin at the Weizmann Institute. So the basic topic here is again 3D, but I'm going to introduce this with something that you've probably seen already at the beginning of this workshop, and that's the head direction coding in 2D in a fly. So you have a fly here sitting on a ball. There are no visual cues available to this fly. Indeed, its eyes are being painted completely black. The fly is free to move its legs, and so the ball will rotate. And what you will see is that simultaneously to the behavior of the fly, you also have imaging smack in the middle of the fly brain in a region that is called the central complex. There is some kind of a ring structure in this region. And you see that the raw imaging data shows some hotspot of activity, some bump. And this bump, if you pay close attention, it stops whenever the fly stops, and it starts moving whenever the fly rotates on the ball. If the fly rotates clockwise, you will see a rotation of the bump clockwise, and vice versa for the counterclockwise direction. Just by looking at this bump, without even knowing what the fly is doing, you can actually decode its heading in the environment pretty accurately. Right? So there is this heading representation that is maintained by a bump of activity, that essentially corresponds to the azimuth of this fly in this 2D kind of world. We have known about this kind of representations long before, sort of having observed this in flies. And the classic observation was a single cell observation. So you can imagine now focusing on one process on this ring, let's call it one neuron. And what you will see is that the activity of this neuron will fire only for specific orientation of the animal. So as the fly rotates, the bump rotates, it will pass on this neuron, and this neuron will, be said to be preferring a specific direction in space. What we would really like to understand is how sort of something like this can enable the encoding of orientation of heads in 3D space. Presumably this is something that is also happening in the fly. Unfortunately, we don't have recordings from the flying flies, but we have other species, as Gilly was showing you before, where this can be done. Right? So that's an example of a bat flying in a dark room, is attempting some kind of landing on a smooth ceiling. It fails on landing on the ceiling, and you see with this sort of graceful recovery is the orientation of the head of this bat is essentially covering a large fraction of the space of possible orientations in 3D. So exceptional navigators, and indeed if you record from some region in the brain of the bat, what you would see, this is a single cell example, it's something like this. So you have conjunctive coding for this cell, for the 3 degrees of freedom that are needed to describe the orientation of the bat in 3D. In this case, it's described in terms of Euler angle, azimuth, pitch, and roll. Right? So now we have a tuning curve, and we really would like to think about the corresponding bump in some relevant space. let me go back to the 2D scenario just to mention how people think about this problem. Sam mentioned models of this kind yesterday. I will go back and forth between 3D and 2D. 2D is just easier to give intuition of what happens. So the idea is now you want to build a network model that essentially encodes a bump that can describe the heading of this fly in 2D, just the azimuth, just the yaw variable. So what you typically do, you consider the manifold where you need to encode this particular variable, in this case it's a ring, so you just follow the heading of the fly as it rotates around. You ideally fill this ring with neurons, and you set up some connectivity structure, so you say nearby neurons excite each other, and far away neurons are inhibiting each other, so that you have some kind of winner-take-all dynamics that creates some localized hotspot of activity that is silencing the rest of the network. So you have some kind of symmetric system, you have symmetric coupling, and you have solutions that breaks this symmetry. So classic model. Now you want to do the same in higher dimensions, and let's consider a simplified intermediate scenario, not the full 3 degrees of freedom required for 3D, where we have a pencil. Now it's very thin, so we just need to consider rotations around its center of mass like this, and ignore the rotations around the long axis of this pencil. And so we need to follow the tip of this pencil. We describe a sphere, the surface of the ball, and you might be tempted, as we were, to sort of parameterize this particular manifold in familiar ways, like this. So it's a latitude and longitude, and create a representation like the one I described for the ring on this particular parametrization that you chose, which is maybe what the brain is doing, but there is a problem. I smoothly rotate my pencil in 3D, and by accident I go close or at the pole of this representation. I will have something smooth happening in the world and some jump occurring in my representation. So this again, it might be that this is something that happens in the real brain, but we would like to have some sort of principled way of create one-to-one smooth mapping between the orientation of the head and the representation in our brain. And so, but the obvious solution to do this, you realize that you have a sphere, you fill it with neurons, you somewhat connect them in a way that creates local excitation, long-range inhibition, and generates some symmetry breaking in this space. Now, what we would really like to do, and surprisingly nobody has done yet, is to do the same thing for the full case of 3 degrees of freedom, like for the case of the bat here, or this kind of 3D Tetris piece that I'm showing up there. Again, we might be tempted to parameterize things in terms of familiar stuff, like azimuth, pitch, and roll, but the strategy would be to actually respect the manifold underlying this the space of orientations in 3D. So what is this space? Now I'm going to say a number of obvious things. So mathematicians in the room would probably be extremely bored. Maybe also other people will be bored. So tell me if I need to speed up. How do we describe the space of possible 3D orientations? One way to think about it is to have some kind of reference orientation of an object, such as this particular orientation. And now we can describe any other orientation of the object starting from that reference just by a single operation, which is a rotation around a certain axis with a certain angle. So, like this, I would have a knife to wield around, so I will just use this kind of silly animations to show you things. You can do this for arbitrary orientations, that's the Euler theorem, but you can, don't need to prove it. You can easily convince yourself that that's the case. So now we need to think about the space of possible 3D rotations. That's a rather complicated piece, and sort of this is one way in which I can draw things on the slide, and but I will need some imagination from your side to actually think about the space. So now we have 3 degrees of freedom, and let's consider a ball, right? So natural 3D ball, we also consider points inside this ball. The origin of this 3D ball will correspond to the reference orientation of the object that we are considering. Any other point in this ball will tell you the 3D rotation that you are using in the following way. This point and the origin will lie on an axis, and this will tell you what is the axis of rotation that you're using. And the distance between these points will tell you what is the angle of rotation that you are using. So if you are moving your point along the z-axis, you will see rotations around z of increasing angle until you hit the boundary of this ball. You can do the same thing, let's say, around the minus z axis, so you will see rotations around z in the opposite direction, until again you hit the boundary of the ball. And now you see that since you can rotate an object by 360 degrees before going back to itself, essentially you have 2 antipodal points that correspond to the same orientation of the object. And this is true for any axis that you might want to consider, right? So the space of possible 3D rotation, you need to imagine this ball with all the antipodal points identified. So it's a projective space, which again is a well-known thing, even though I don't know who is the first person that realized that this was the manifold underlying 3D rotations. Maybe you can tell me later. But the problem is we didn't even know how to write functions on this space. But luckily, mathematicians solved this for us some time ago in the 1920s. And the idea is the following. I'm going back to the scenario of 2D. So now we go back to the ring. And people, when thinking about problems on this ring, again, Sam showed something related to this yesterday. You have some function on this ring. This might tell you, for instance, what is the coupling between neurons as a function of the difference between their angle on the ring. And since it's a function on a ring, you might be tempted to just write this function in Fourier series. So you have some constant on this ring that usually describes the long-range inhibition that you have in your circuits. Then you will have the first Fourier mode that essentially will tell you, well, if I have nearby neurons, I will have strong connections, and you might have additional modes, and so on and so forth. Right? And what people do, what Heimson-Politsky did is essentially say, I stop here, I just consider the first 2 modes, it gives me my local excitation, lateral inhibition. I have 2 parameters describing this coupling, and I can tell you anything you want to know about the solution of a neural network that has this particular coupling. So, we would like to do the same on this 3D manifold. And the key to make this jump is to think about this Fourier decomposition in a slightly different way. And so we have our Fourier modes that are indexed by some integer, k here. And one way to think about these Fourier modes is now in terms of them being a representation of the group of rotations in 2D. You heard about groups, I think in the Monday morning session. So that's a one-dimensional representation, a linear operator that acts on the 1D space. And essentially, the key point of this representation, it needs to respect the composition relations that you have in your group. So if I take 2 group elements now, 2 representation, one rotation of angle theta and another rotation of angle theta prime, I combine them, in this case by just multiplying them, and I obtain something that should be the composition of these 2 in the original abstract group, which would be a rotation of angle theta plus theta prime. So just to understand where you're going. So you want to build a network model for this rotation space, but such that the representations or the codes satisfy some, you can easily compute the composition of 2 rotations, or like why are you? I just want to write function on this manifold of rotations. This function can describe coupling between neurons. They can describe the activity of the neurons themselves. And one systematic way to do it is, for the case of the ring, using this decomposition. And this decomposition turns out generalizes to more complex group, not just the group of rotations in 2D, but any Lie group, any compact Lie group, which includes the rotations in 3D. So the strategy is to just set up a coupling according to the generalization of this, and then study the resulting network solutions. Does it make sense?
I don't know. Well, I hope.
So another way to look at this is now thinking about a representation of the groups corresponding to this ring manifold. I have a representation indexed by my 0, k equals 0. It's just a constant, again, my inhibition. And then I have, my first Fourier mode, second Fourier mode, etc., etc. And these are just representation of groups. And as I was saying, this generalizes to more complicated groups.
Are the A's down below there real, complex?
Real. I'm considering real functions here because I want to describe things in terms of neurons and synapses.
Okay.
Neurons have real activation and synapses have, real synaptic weights. So that's going to be a slight complication in that it's much easier to deal with representations where in complex space, at least for the case of the rotation group in 3D. So there's some work to do when you want to work in real space. But so this now generalizes to any compact Lie group, which includes the group of rotation in 3D. The key difference is that, as you all know, rotations in 3D do not commute, right? So if I have two rotations and then I switch the order, I obtain a different end result. And what happens when you have a non-commutative group like this is that now your representations are no longer guaranteed to be one-dimensional, so the nice kind of Fourier decomposition that we all know and use. And so now your basis function will become higher dimensional. It will be matrices. And for this particular group, it just so happens that you have the first representation here will still be one-dimensional, like my flat inhibition in this space. The first non-trivial representation will be a 3x3 matrix, like an actual rotation matrix, a faithful representation of this group. And I need to consider linear combinations of the elements of this matrix, so that's what this trace is doing here. And now this A1 is an actual matrix, and then it grows like it goes 5x5, 7x7, etc., etc. Now we have a way to write couplings on this manifold, and like Sokolinski did, we stop here. We have our global inhibition and local excitation. But now we have a lot more freedom in sort of setting up a system that creates bump of activity anywhere on this bizarre manifold. Because before we had two scalars describing our coupling, and now we have one scalar, one 3x3 matrix. These matrices need to be symmetric in order for the bump to be stable and not just move around spontaneously. So we have like 7 parameters that we can use to set up our network. So a much richer kind of set of solution. This was unclear and probably was, I apologize for that. What we did, what Hervé did actually, was to find sort of a systematic way to consider this manifold, fill it with neurons, generate coupling that is local excitation and respects the topology with the antipodal points identified, and write down equations for the network dynamics and actually solve it and understand, at least in some cases, for some choices of this particular matrix of coefficient described in the first mode, sort of understanding which kind of solutions you have. And I'm not going to go into the details of that because that's going to take a while otherwise. So essentially what you have at the end of the day, this is one solution corresponding to this coefficient matrix being proportional to the identity. So it's one of the simplest. You have your ball of neurons here, each dot is a neuron. I'm just showing you the 2D projections for clarity. Color and size will tell you the firing rate of these neurons. And what you see is that you have some bump that is localized in this particular topology, and this bump can sit anywhere. So what are we going to do with this? Now, what we would really like to do is make some contact with that data. So far, this is a little bit abstract. And so that's an ongoing work. One thing that we would like to understand is whether some of the solutions that are coming from this model, and we are still studying them, we are still classifying them, maps onto the kind of heterogeneity that is being observed within a species and across species in the coding of orientation of head in 3D. So the simplest thing that we can do is just assume that perhaps this is the solution. And maybe we can visualize this solution, this tuning curve, in whatever representation people are sort of familiar with, like azimuth, pitch, and roll. Here I'm showing you a 2D projection of this. And so you can imagine, since these are two distinct manifolds, then you're kind of trying to map between them. Something that looks nice here will be somewhat distorted and slanted in this representation. And the hope is that sort of this kind of activity patterns can explain the actual activity patterns that we see in glial cells. On the other hand, we also know that the heterogeneity is not just reflected in sort of weird firing patterns when looked at in let's say, azimuth, pitch, and roll. There seems to be a coding that is not just reflecting the 3 degrees of freedom that are necessary to encode the orientation in space. There are lots of cells that care only about 1 or 2 degrees of freedom here. So that's a summary statistics from experiments in bats from Nakamura-Noski. And the idea here is that essentially by choosing appropriately the matrix of coefficient that describe the way in which neurons interact, we can sort of create neurons that have this kind of reduced coding. Like, for instance, what they observe for cells that code azimuth is something like this. So when you have a bat crawling on the ground, you will observe in a cell a certain preferred heading, and when now the bat crawls on the ceiling, you will see that this preferred heading is actually shifted by 180 degrees. Right? So if we consider one possible coupling in our network and look at the corresponding solutions, we can observe sort of features that map onto what Nakamura-Noski observed in his bats. There are other observations coming from rodents, where actually there is so-called tilt azimuth representation. So what you have here is now a rat that is allowed to climb on walls, and so we climb on this wall, west, what is this? It's called south and east wall. And the question that, this was Kate Jeffrey work, the question that she asked is how does the representation of tilt azimuth, which is how does the representation of heading on each of these walls with the common reference being the zenith here, how does it change when the rat moves from one wall to another? And you see one representative example here, where you have a certain heading preference when the rat runs on this west wall, and as it goes to the adjacent wall, this heading preference sort of rotates by 90 degrees, and it does so when crossing to the other wall as well. We can find other solutions in the model that actually have this kind of behavior as well. Okay, so we haven't characterized all the solutions. I just showed you that some of the heterogeneity that we observe in the data can be at least accounted for by some network structures that we can generate with this kind of models. But why would we have different networks even within the same animal? And this can sort of, I think, be explained by the fact that this particular system is performing a computation. And this computation is essentially integrating proprioceptive information in such a way that the bump is location on whatever manifold, the ring in 2D, and this projective space in 3D. The bump is updated, the bump location is updated to actually represent the current heading. So you are integrating, let's say, angular velocity information coming from the legs of the fly, for instance, in 2D, And for doing these kind of computations, then what you need to do, as other people have shown, is to sort of have more of these network models. For the case of the two-dimensional system, this is one particular example that I think was originally conceived in the McNaughton lab. And what you do is, instead of considering just one ring where you have your pump of activity, you consider two additional rings. And you also have pumps here. And so you have couplings from this, let's call it central ring, to the side ring in such a way that the pump sits at the same location in all three rings. But now the connections back from this side ring to the central one are a bit funky. So they are shifted in some way. And if I look at the left ring, you will see that the connections are shifted in this counterclockwise direction. If I look at the right ring, it's shifted in the clockwise direction. So they want to exert a pull, they want to move a pump. This one wants to move the pump this way, this one wants to move the pump this way. If they are both active at the same level, this pull will cancel and the pump will be stable. For now, imagine that these two side networks receive angular velocity input, just a scalar that tells how fast I'm rotating in space. Then one of these networks might win, and it will pull the pump accordingly. And if you set your connections in the proper way, then you can obtain something that is very close to linear integration, right? So a linear dependency between the phase of this pump where it sits and the angular velocity input. Now we can do the same in 3D, just for 1 degree of freedom. We can imagine now that we have 3 of our networks. There are similarly shifted connections between the side networks and the central one. So now we can do integration into with this system. But of course, we would like to do so for any movement in 3D. And what we need to add is essentially additional networks that take care of integration along 2 additional axes, right? So in this case, this is rotation around the z-axis, and we need 2 additional pairs of network, let's say, for rotations around x and y, and however many generators we have in our group, essentially. So, right, in this architecture, if the network needs to perform angular integration, you need to have many networks that in order to do so. And essentially, this suggests that you might have heterogeneity in the connection within the network that can create different tuning curves within an animal, and perhaps different solutions when you look at other animals. So one question is whether there is some optimal configuration of connections, of structures that can perform angular integration, and maybe this would suggest why we see exactly what we see in this species.
Yes, so if you would set it up like that, wouldn't that be doesn't that relate to whether the tangent bundle of your SO3 space is trivial or not? So I mean, you kind of look. I don't think you can do that in a consistent way. I mean, the nice thing that you can...
we can do in a consistent way, meaning that, we can build this and actually integrate things, right? So you are essentially...
Will you be able to wire it up consistently at the boundary? I get it on the interior there, it's just a free ball, but...
Yeah.
Anyway.
At the boundary of what?
I mean, you got a 3-disk with your you quotient out the antipodal map on the boundary, right? Yeah. So but this space won't have a trivial tangent bundle. That means you can't find a consistent way of telling at every point. You can't assign 3 fixed directions at any point such that it's consistent. Anyway, we can talk about it later.
I can ask a more elementary question along the same lines.
Just a second. I don't know if is this related to sort of thinking about a vector field on.
Yeah, I mean something. You wouldn't find 3 orthogonal vector fields non-vanishing on that space, I think. But we can talk about it later. Sorry.
Yeah, I think you can do it for SO3. This wouldn't work in other.
Oh no, it's trivial. Yeah. Sorry, yeah,
this wouldn't work in other groups.
It's 3 dimensional.
Here you can definitely do it. Now you have a lot of freedom in how you want to set up these particular interactions, right? But we know that this works at least because we've actually constructed a realization of this, integrated kind of vestibular input and update the bump in a correct way. The question for us is, since there are many ways to do this, what's the best one?
Yeah. If your blob of active cells goes up to the edge of your sphere, does the dynamics you've created automatically result in it coming back from the opposite side?
Yeah. That's automatically.
Doesn't that require fairly long-range connections of a specific kind?
Doesn't that
Require long-range connections going all the way across the thing of a specific kind?
So I should specify that this nice arrangement of neurons in the flight on the ring, that's really some kind of lucky accident.
No, I'm not talking about the fly, I'm talking about the ball.
Yeah. But what I mean is that you shouldn't be sort of imagining mapping between the functional space, so the heading preference of these cells, and cortical space. Not the one-to-one mapping, right? So, in other words, for the case of the fly, let's think about the 2D scenario with the ring. When you look at the rodent data, you see cells in certain regions of cortex and also subcortical area where you have heading preference there. But there is no anatomical structure that maps onto the functional preference of these cells. In other words, they are just shuffled around. If you have a ring like this.
I'm just wondering if you understand my question, because
Yeah, if you have a ring like this. Okay, now you have neurons like arranged in this way, and you're asking, you essentially need, when you use Fourier coupling, right? For the first mode, you would need long-range connection, meaning that this neuron needs to
the
connect
question
to
I've
this
asked does
neuron.
not apply in 2 dimensions.
It doesn't apply there.
It doesn't apply.
Oh, sorry, then I definitely didn't understand.
No, you pointed out that the sphere has opposite antipodal points that have to be identified.
Yeah.
Does your actual dynamics do that?
The dynamics does that.
Then I'm sort of interested in how, but you can get there eventually.
It does that. Okay, so for the dynamics, yes, it does that, and it does that because essentially it's built in the way in which we describe the coupling between the neurons.
Okay.
Okay, so, sorry. I
But
thought you were asking.
that describes the coupling between the neurons. I guess I don't see how it describes the coupling between the neurons.
This is telling you essentially. If I consider a neuron sitting at the identity of this group, this will tell you what's the strength of the connection to another neuron that encodes another group element. And now you can sort of extend this to other neurons that are sitting anywhere in a trivial way. In other words, when you look at the connectivity between two neurons, one encoding G and one encoding G prime, you will have that your coupling will be essentially this function evaluated at G times the inverse of G prime. This is what
So
the actual
there's
connection.
one real valued function on the sphere or the ball
Yeah,
that actually defines all the couplings among all the neurons
Yeah.
in that ball.
yeah.
Okay. Don't see that, but okay.
And so that at this point you can define your dynamics and your bump of whatever shape you can generate, right? I can move it in a way that you will just
Okay. Thank you.
jump back here. So the dynamics, sorry. Okay, so this is one potential way to sort of think about the heterogeneity that we have in the data. Again, analysis of the data is ongoing, so I cannot tell you much about the specific relationship between this model and what happens in the actual brain. But in the last few minutes, I would just like to discuss something that is now related not to the orientation of myself in 3D space, but the orientations of objects in 3D space and the way in which humans process sort of information related to the orientation of these objects. There is a famous experiment in the 70s from the lab of Roger Shepard. How many of you have have seen this experiment? Okay, that's less than half. So I will just show you quickly how this works. So, like participants in this experiment, essentially you are looking at two objects, 3D objects that are projected on a 2D screen. And all you have to do as participants, you have to tell me whether these two objects are the same up to a rotation in 3D. Just raise your hand if you think that's so. I have to wait a certain time and you will see why. Okay. And just give you another couple of trials so that you get a sense of what happens here. Just raise your hand if you think that it's the same. Last one. I know that I have to wait a certain amount of time. So that's fantastic group of participants here. So this is considered 100% performance. The first one was correct. The second one was same. Second one was same, and this one was not the same. And what Shepard did was to go to each of the individual participants and ask them, how do you solve this task? And so the introspective account of these subjects was invariably more or less the same, which is I just focus on one of the images, or maybe a sub part of the image. I rotate it in my mind and then I see whether it matches or not with the other. Right? So that's the famous mental rotation experiment. Introspective accounts are not so interesting. The cool thing about this psychophysics experiment is that he found an actual correlate of this introspective account. And he did so by essentially measuring the reaction time it takes the subject to tell whether two objects are the same as a function of the angle that is needed to make these two objects congruent through a rotation. What you see is some kind of striking relationship between these two quantities, somewhat suggesting that if you need to tell whether two objects that are 120 degrees apart are the same, you go through intermediate steps, right? This mental rotation story. Now, intriguingly, this is true regardless of whether the rotation you have to perform is on the plane of the screen, like this data, or off the plane of the screen, like this data. So you see a very similar relationship, suggesting that all that matters is the angle between this object and not the axis of rotation that you need to use to make them congruent, which is a very intriguing observation for us because of one aspect of these classes of models. And now I'm going to model this in a very dumb way, so I apologize for this. Please.
Quick question. What if you have to do the rotation along both axes at the same time? Like if it's diagonally through the plane.
That's another, that would be another axis, right? So you can always do it with just one rotation. And not in these experiments, but in other experiments, they showed that
it's the same.
You see a similar relationship, yeah.
But
you shouldn't be thinking about the two axes simultaneously. It's just another rotation with some different axes. I think at least that's one way.
But you also have to find the axis somehow.
You need to find the axis, right? That's the computation, which is kind of cool. And I'm going to model this computation as I was saying, in a very dumb way. So we know that some of this might be done by a visual system, and now we perceive local patches in D4, TEs, and the tons of the things we have seen. Now I'm going to assume that this computation can be conceptualized just by having one single network that encodes the orientation of the objects of interest.
I believe that Georgiopoulos and Andy Schwartz did this and autocortex also.
Yeah. Mental rotation of the population vector for action. This was done in 2D, I believe. They did the same thing. So the model is extremely simple. What I'm going to do, I'm going to assume that we have one such attractor network for bumps in this 3D rotation space. And what we are going to do, we are going to, when I say that we show one image to this network, it means that we are going to bias with an external current a subset of neurons that care about the specific orientation of the stimulus in 3D space. And so a bump will be formed there. And now when I show another stimulus, again, what I'm going to do, I'm going to bias a subset of neurons that now code for the orientation of this one, right? So this is super dumb. I'm giving this network a lot of information. I'm essentially telling it what's the orientation of one of the stimuli to set up the bump, and what's the orientation of the other stimulus. And so the network now needs to move the bump from here to here. The interesting computation is that in doing so, it discovers what's the axis of rotation that it needs to use. Because essentially the shortest path in this particular space is one that fixes the axis of rotation and just increases the angle. It's a subgroup, one-dimensional subgroup that discovers the axis essentially. So if you run the simulation for one particular pair and you map back the location of the bump to the orientation of the object, you see that this is a finite simulation, so there is a little bit of noise. But what you see is that the network is discovering the axis of rotation and just changing the angle. And if you do this kind of exercise for very many pairs, then you can measure in a way that's similar to what Shepard did. You can measure the reaction time of this network, right? The time it takes for the bump to move, versus the angle of rotation that is needed to make the two objects congruent. And you find sort of this linear relationship. And you might be tempted again, as we were in sort of building models of orientation coding of the head in 3D, to use another parameterization, perhaps Euler angles like azimuth, pitch, and roll. And you can play the same game there, and you would see something that is sort of strikingly similar between the two views. Now, sort of the distance between 2 points in this particular space depends on the axis of rotation as well.
Why is it more noisy?
It's more noisy. It's more noisy because for a given angle of rotation, I'm using several different axes, and now these several different axes will have different distances. The path that the network will choose, which depend on the specific axis, and this contributes to the variability that you see here. So with this, I would just like to sort of conclude. This is again a very simple model. It produces though a rich class of solutions, and so our aim is to characterize the taxonomy of these solutions and map them to the zoo of direction neuronal responses that are being observed in rodents, bats, and so more recently also humans, at least in fMRI studies. One open question is, how to perform angular integration in some kind of optimal manner with these kind of models. And in thinking about these psychophysics experiments in humans, we are now really becoming interested in understanding how and whether and how these kind of networks might contribute to our perception in 3D space. And we are collaborating with the lab at Geneva, where essentially we are showing these 3D Tetris species to mice while imaging the activity of neurons in the entire cortex to try and understand whether there is some relationship to 3D features, maybe locally or globally. And so from the modeling side, we would really like to make this model a little bit more complex and sort of use more complicated groups of transformations like projective transformations. That's one thing that people in the computer vision field do quite a bit for a number of reasons. That's one example from a computer vision book where you look at this kind of painting, and essentially what you would need to do is somewhat imagine what the configuration of this room would look like if you were looking at it from this particular perspective. Right, so this is something that you could probably do in your mind, similar to the 3D rotation experiment, and you would do a pretty good job at that. With computer vision, you can do an excellent job. So we would like to understand secret mechanisms that might underlie these kind of transformations with similar approaches. And with that, thank you so much for your attention. Again, this is work by Hervé and Alon Ruby, Marseille and Weizmann Institute. And I want to thank Arseny Finkelstein and Nakon Planowski, as well as Jean Laurent and Dora Angelaki for lots of interesting discussions about this kind of codings in bats and rodents.
How does this network model account for non-commutativity of the rotation?
What do you mean by
Well,
accounting
I
for
thought that
non-commutative?
the model is that you have a kind of a neural network, and the bump from one rotation pushes it one way, from a different population pushes it in a different direction. But then if I add signals from population 1 and population 2, it will be independent of the order in which I add, you know.
Yeah,
And
so
then
that,
the rotations should, the results should differ for rotations, but in this model, the result will be the same.
So these pairs of networks essentially tell me what's the angular velocity when I
No,
rotate
when-
around the specific axis, and now all I need to do is integrate this. And so if I mimic two rotations in a sequence, I will have a pump moving somewhere that encodes for my 3D orientation. And if I switch this sequence in the inputs, I will have my pump going somewhere else. It's sort of consistent with the non-commutativity of these kind of operations. That's
So it
probably not.
changes the radius. So the input of this pair will change.
I have a bunch of scalar inputs coming here, and now I can define two sets of scalar inputs corresponding to two distinct rotations, and apply these inputs here. This central network will integrate this information and move the bump accordingly, right? And depending on the order in which I present these scalar stimuli to these networks, my bump will end up in one location rather than another one, consistent with the non-commutative properties of the group.
Maybe, yeah, maybe later.
Okay.
Let me take one more question and have a break. So you
What in your model? What sets for the mental rotation task? In your model, what sets the time scale? And I guess what I'm also wondering is clearly that relationship which links time for angle must be non-monotonic because eventually it must turn around, presumably.
Yeah. That's absolutely. So in the actual experiment, this thing goes like that. When you consider right the
Yeah.
entire range to
Okay.
360, so they are just
And-
mapping it back.
And what sets that time in your model? And what can you say about that in the context of what Shepard and Metzler measured?
Oh, that's a good question. There are lots of things that contributes to the actual speed of rotation of the bump in the model. One is the
What's the
time
slowest
constant time. Okay, the slowest one is probably determined by how much overlap do I have between my actual bump of activity and the input with which I'm driving the network. I'm not sure it's a completely real relationship.
All right. Let's thank you again. You guys can keep talking during the break.